Welcome, everyone. Today, we are understanding the Indian identity and our society through genetics with Dr. K Thangaraj, who works at the Center for DNA Fingerprinting and Diagnostics at Hyderabad in India. So our first question would be that in the past decade, history as a discipline has seen a revolutionary change in terms of the research methods that are used. So an interdisciplinary dialogue has been established with biology, especially genetics, facilitating DNA fingerprinting and mitochondrial DNA areas of your expertise. And these techniques have been used to analyze and trace migration patterns and population history. So situating this to an Indian context, what is some of the ongoing research and how does it tell us about Indian biological identity?
Yeah, thank you very much for this question. And as you have mentioned that this aspect has been going on for a very long time, particularly to understand population history. It's kind of multidisciplinary, people with a different background, historians, linguists, anthropologists, archaeologists, now geneticists. We all work together to understand or work independently, work together to understand this very interesting issue. Where most of the other field, for example, if we take history or language, something, because those are not directly can tell about what was our past, right? Because it has been returned somewhere and somebody follows and transmit to that next generation or whosoever is being following that. Whereas DNA studies, and even without any information, one can be able to study the DNA mutations and able to connect people all over the world. Right? So whatever was there, not today, not 100 years back, 1,000 years back, several tens of thousands of years back can be traced by using. And in one of our early study, maybe more than 20, 25 years, that we have studied the populations of Andaman and Nicobar Islands. There are 4 tribal groups who typically look like Africans: Onge, Jarawa, Great Andamanis, and Sentinelese. We studied 3 of them, and we traced back that they are the first modern human who migrated out of Africa. And we also calculated the age based on the mutation or change in the DNA and estimated that probably they reached Andaman and Nicobar Islands about 65,000 years back. Okay. So that's initial human migration from Africa. Right? And while migrating, probably some group stayed in southern part of India. And further they migrated to Andaman Nicobar Islands, from there to Australia. Okay. Even today, people are connecting Australian Aboriginals, Indians, some group in India and Africa. So that tells that there's a migration. And subsequently, what we have studied is that and identified in the prehistoric India, probably there were 2 founding groups. One is ancestral South Indians, who are part of the early human migration. There was second wave of migration brought people to north, and these 2 founding groups gave rise to many population groups. And now what we see in India is the mixture of these 2 founding groups. So that mixture happened about 2,000 to 4,000 years back. Okay, initially 2 founding group, they gave rise to many populations, and there was admixture between those 2 groups. And now, after maybe last 2,000 years, after admixing, every single population is maintaining the endogamy, means marrying within the group. Because for 2,000 years, every single population is marrying within the group, the DNA changes happen in one group may not be the same in the other group. That's why every group looks very unique in terms of their genetic material. Okay. And if you see within India, you don't see any group without admixing. Okay. So that you can obviously see with many characteristics, like our color. Unlike if you go to Europe or East Asia, like people look similar with their skin color also will be very uniform, right? But in India, we see range of color, very light color, very dark color, in between so many colors. So it's all kind of indication that the people have admixed within India. So this is the original settler or the ancestral population. In addition to that, there are several new waves of migrations, like the Jewish population. There's a population called Siddhis who have been brought very recently, few hundred years back. Parsis, right? Like that, there are several migration into India. And now if you look at India, it's kind of full of diversity. We have more than 4,600 anthropologically well-defined population groups. Anywhere in the world, if you go, you will not find such a huge human diversity. So that's our strength, not only in terms of genetics, you know, the culture, language, everything is very diverse, right? So if you say what is the identity, we cannot say that one particular identity, right? It's a kind of very diverse, you can say that is the identity of India.
Thank you for that answer. Moving on to the next question. Interdisciplinary research demands new kinds of research methods used. In this case, the research question is inspired by a historical problem, but the methods employed to explore the problem are biological. Can you elaborate on some of the techniques you use to explore population genetics in India?
We use different techniques. If you wanted to trace the maternal lineage, we use the mitochondrial DNA. Okay, because we all inherit the mitochondria only from the mother. There's no contribution of mitochondria. You know that it's a powerhouse of the cell, which comes only from the mother. The father's mitochondria in the sperm, it will be in the mid piece of the sperm, so that will not get integrated at the time of fertilization. It remains outside. Of course, there are some rare cases. Those rare cases always cause some disease. So we all inherit the mitochondrial DNA only from the mother. So mitochondrial DNA is very small, circular DNA, 16,500 base pair. And that is used to trace the maternal lineage. Okay. But if you want to trace the paternal lineage, we use the Y chromosome markers. Okay. But of course, that can be studied only in the males. So we have 46 chromosomes. 44 chromosomes are common between male and female. There's no difference. In case of male, there's one X chromosome and one Y chromosome. Okay, Y chromosome is the one which carries the gene which makes the individual as male, like S or Y. Okay, whereas in female, there are two X chromosomes. The Y chromosome is exclusively present only in the male. So we wanted to trace the male lineage, because the Y is being transmitted from grandfather to father, father to son, son to grandson, so on. So we want to trace the paternal lineage, we use the Y chromosome. Now, because the technology has changed, right, the whole genome was sequenced, and more than 1 million marker can be analyzed in a single experiment. Okay, so there, the combination of both markers which transmitted or inherited from father, from the mother, can be looked at. So 3 different kind of markers are used for any population genetic studies. But in case these are for contemporary sample, if you take the blood sample and a few months old, still you can use. If the samples are very old, thousands of year old sample, we cannot use the routine methodology. We have to use the ancient DNA technology. That's in the ancient sample, you get very small quantity of DNA, particularly the intact DNA. DNA would be highly degraded, fragmented. So we want to use, and also there could be a contamination. Somebody who's, if they're not handling sample properly, for example, if I am using, taking the sample, but my cells, some of the cells, the epithelial cell, if I am talking, right, there could be a possibility of contaminating that with the ancient material. That would amplify and give us the report of the contemporary individual who is analyzing his sample. So therefore, that has to be analyzed in a specific laboratory called ancient DNA laboratory. That is always maintained with the UV light. There's a EPA filtered air comes in, right? So much of kind of sterile condition, so that the DNA which is of modern would be degraded or fragmented. Therefore, we won't amplify that. And there are also several methods, because not every single sample can be used with the same method. Because of the nature of the sample, we have to alter the technique to, and also use different kind of protocol for amplification. So it varies from sample based on the nature of the sample, where it has been obtained and so on. So that, of course, ultimately we use the PCR and sequencing. We amplify smaller fragment because the DNA is already degraded. And sometimes we use the whole genome amplification and sequence the whole genome. So the technology is being evolving, and we also adopt different kind of technology, and we use based on the requirement, based on what we are going to address, right? What is our objective of the study? So in that case, it varies from objective to objective. Otherwise, these are the overall techniques which we use.
Thank you, sir. As you said in your very first response, that in India, we don't have a particular identity. We always have identities. So you used plural. In that sense, cultural history are also plural. We have a lot of cultures. We have a lot of history, and that is the strength of India. But these cultures and these histories, they also often inform identities. As you said, the present political climate suggests that a singular national identity is also taking shape. So how do you see the meaning of India itself changing with this new genetic testing that is happening? And we are identifying different migration patterns. As you said, 65,000 years ago, the first modern humans came to Andaman 65,000 years ago. How do you see India right now changing because of these kinds of techniques in place?
I don't think India is changing much because of the techniques. We are only understanding what is the reason for such a huge human diversity, right? Using the technology. Otherwise, until very recently, we use, if you look at how the population genetics were studied, people used to use the blood group, because obviously very few number of blood groups, so you cannot actually differentiate the people of the population. Then the protein, serum protein polymorphism, again we have very limited number. Then the mitochondrial DNA was used, Y chromosome was used, and now the 1 million SNP array has been used. Now the whole genome sequencing has been used. So that's the ultimate, right? So when you use the whole genome sequence, and if you are putting a question that about the identity of one particular individual or one particular group, then you have much more information to connect with other population. So you can see that which are the population which are more closer among the 10 particular population you have analyzed, right? So in that sense, that the technology helps us in understanding and come to the conclusion in much better way than the previous time. Okay.
And so,
Did I
also,
answer your question? Or
yes, I just have a follow-up question to that. How is this
yes.
also changing the Indian research landscape, particularly in population genetics? Because we are looking at a lot of new techniques as well as a lot of new people coming into research, because there is a lot of excitement going on around population genetics, specifically the Rakhigari site in Haryana, understanding the Harappan population and everything. So how do you see the current research landscape growing? And possibly say even growing in the next 5 to 10 years?
Yeah, if you look at the population genetic studies, it's very, very important because it can address several issues and give clue to many issues. Right? So we cannot see population genetics just to understand only the population history, because it also has a lot of offering to the medical field, right? So I can tell you some of the examples from my own study. So we studied large number of populations to understand their population history, not only in India and also other South Asian countries. Then we predicted that lot of population in India or South Asia, at least 1/3 of the populations, are having a population specific recessive mutation, which may lead to population specific disease, or even we can call it as rare diseases. Right? I mentioned earlier that because populations are marrying within the group, okay, for the last 2000 years at least, if there is a mutation change happening in the non-coding region, of course, that's not going to cause any phenotypic change or obvious disease and other things. If it happens in the coding region of the gene, and that mutation, again, you can divide the diseases broadly into two categories. One is the dominant disease, other one is recessive disease. As I mentioned, we have 46 chromosomes, or you can say 23 pairs, right? 23 chromosomes we inherit from the mother in the form of egg, 23 chromosomes we inherit from the father in the form of sperm, right? That fertilize and become 46. So that means every individual, half of the genome comes from the father, half of the genome comes from the mother. Okay. In the case of dominant disease, if there is a mutation in one of the pairs, so I said 23 pairs, right? If you take chromosome 1, there are 2 chromosomes 1, and 1 chromosome carry the mutation. It still shows the disease. That's a dominant. If it is a recessive, if 1 chromosome is affected, 1 other chromosome is normal, then it won't cause the disease, right? But the person is carrier. For example, if the mutation, such mutation, recessive mutation, arose maybe 10 generations back, for example, in one of the individuals. At that time, there was only one family settled in a village, right? Now, they have expanded, and the mutation which supposed to cause the disease in when there is a 2 chromosome having the same mutation, right? Right now, initially started with 1 chromosome with the mutation. So that is going to well spread within that community or within the initially family, the family then become community, right? When it happened, as long as the individual carrying the only one mutation in the pair, it's not going to affect. But it's expanded in the population. Now, the boy and girl are with heterozygous mutation. That's 1 chromosome is affected, other chromosome is normal. If they marry, right, and there's a possibility that they can contribute transmit the abnormal chromosome, both the father and mother, to the next generation. But the child carrying the mutation on both the chromosome will have the disease. So that was the prediction. Right? Now we are seeing the large number of such population. We identified that the children with the disease carrying the mutation on both the chromosomes, whereas the father and mother are carrying the mutation on the only one chromosome. Right? So we predicted 1/3 of the population. Now we are actually seeing some of the populations are carrying. So we are expanding the study to identify the mutation in the large number of such populations. So that would help as in first identifying the mutation, then do this population screening, right? Then give the counseling to all the families who are carrying the mutation. We can say that you can go for prenatal diagnostics or even the premarital. If you identify the mutation in few individuals, you can say that, okay, this is the thing is going to cause this disease, and you should not marry the person who carry the same mutation. That would in fact help in just stopping that mutation further going to the next generation. Therefore, that disease can be eliminated if you do very systematic analysis, screening, counseling, and so on. So that's the kind of thing one would expect. And in fact, this would be the population genetics would help in many ways. This is just one example. And even same thing in terms of infectious disease. You can say that there are certain population carrying the mutation which makes the individual susceptible for the disease. Several examples from our own study, like malaria, leishmaniasis, even the rheumatic heart disease. We found that the people with the mutations are having the disease, whereas the mutations are not having the disease. So in that sense, population genetics very, very important in the future, maybe 10 years or 20 years, or even more than 30, 40 years, because this will continue. This will help as in going at a population level, screening, counseling, everything would be based on the population genetics.
As we've already talked about when we foray into a subset of your research in the paper, Reconstructing Indian Population History, you've said that India has a wider allele frequency between groups compared to Europe. Therefore, as you have said that India has a more genetically diverse population, to what extent do you think the evolutionary history of the present Indian population impacts the disease patterns? For example, one of those is our predisposition to diabetes and the overall health quality of Indians.
I think the one which I mentioned earlier, that what we have is that population groups in India, there are 4,600 populations, and every population is practicing the endogamy. Because of that, allele frequency of a particular mutation would be, if that is causing the disease, would be very high only on that particular population, not in the other populations. So if you see overall a particular disease, it is a population specific in nature, the allele frequency will be very high in that particular population. If you look at the allele frequency of Indian population, it will be very, very low. Okay. But if you take some of the mutations which whose allele frequency is very high across India, that means that particular mutation must have arose very, very long back, much before the population diverged. Okay. For example, I said the Indian population is approximately 65,000 years old. Okay. If that mutation is more than 20,000 years old, because I said the admixture took place around 2 to 4,000 years back. If the mutation is much older, and you would expect that mutation is well spread across India, which we have shown. So one of the mutation in a myosin binding protein C3 gene, which causes one of the heart disease called cardiomyopathy. Okay. So that disease causing mutation is about 4.5% across India. Okay, that's in the heterozygous condition. Again, the mutation in this case, if it is in the heterozygous condition, that means one chromosome is carrying the mutation, other chromosome normal. Then it shows the disease after 40 or 45 years. But the mutation is in the homozygous condition, that means both the chromosomes are carrying the mutation. Then the disease appears at very, very early age, less than 10 years, which we proved by looking at the postmortem tissue of the children who died due to cardiac arrest. Okay, about 33 children we have analyzed and found 10% of them are carrying the mutation in a homozygous condition. And it also causes, there are three different kind of cardiomyopathy. One is dilated cardiomyopathy, where the ventricle enlarges beyond the normal size. Other one is hypertrophic cardiomyopathy, where the heart muscle become very thick. Third one is restrictive cardiomyopathy, where the heart muscle become very stiff. Because of that, the heart cannot pump the blood, and suddenly it leads to sudden cardiac arrest. Okay. And we estimated when this mutation arose and found that this mutation is 30,000 years old. Right? So because the mutation is very old, and that time people must have been very small in number, and it has been passed on generation after the generation, and because of the population mixture, population migrated, so it got well spread across India. So again, population genetics helps us in looking at how old this mutation is, how it got well spread, and many more things can be done using the population genetic approach.
Thank you, sir. I believe we are going towards the end. So the final question would be, population genetics, as you've mentioned, will be a pertinent field even in the future. But this information will only be useful when it is communicated to a wider population of people, to the entire population of India per se. So how do you see population genetics itself growing in the next 10, 20 years, and the information being disseminated to a wider Indian population? Because right now it is again a very small subset of a field that is still growing. So how do you see that the information being disseminated to common Indian people?
Yeah, so whenever we find something interesting, like the way I was mentioning the cardiomyopathy, and the one where I've shown that at least 1/3 of the Indian populations are probably carrying the mutation, which may cause population specific rare disease. So whenever we publish this in a scientific journal, we also write a simple article and circulate it to the media. Okay, so you can find out that just put some of my study title, then you will find that is the newspaper, right? So even yesterday there was some news, like if you put CCMB and male infertility, you will find there are several newspapers have covered our article yesterday. Okay, so we disseminate our knowledge to the public in a simple language where they can understand, and even some of them comes back to us and ask whether we can do the test for them, whether they carry the mutation or not. Okay, so as a scientist, that's our responsibility, that whatever we do in the laboratory has to reach a common man. Okay, right? Both the way, we go to remote place to collect the sample, right? So in fact, I traveled Andaman and Nicobar Islands and myself collected the samples. right? Similarly to many tribal areas. And we know that how the populations are in the remote place and remote village, and sometime in the remote jungle, right? And having seen them, so it's our responsibility that do the analysis and convey our finding to the public, right? It could be used directly by public, and the public come forward to test themselves whether they have the mutation, or it will be also useful for making public policies. And taking advantage of this information, we go back to the funding agencies and ask, this is our finding, so this is what we want to do further, and we want the funding. Okay. One such thing is that because of all this background which I mentioned, we approached DBT, Department of Biotechnology, to look for mutations for very rare diseases in India. Okay. So rare disease, as I mentioned, that it could be very rare when you look at within India, but if you go to a particular population, it is maybe more common, could be 10%, 20%, right? So this is one of the major problem in India because of our population structure, endogamies, and also consanguinity. So we approached, and there are CDFD is a nodal laboratory. We have another 15 laboratory all over India to do pediatric rare disease program. We wanted to identify the genetic cause. After screening the known genetic markers, we will do either the coding region of the entire genes, that approximately 30,000 gene, or some case we do the whole genome sequencing of some of the rare disease patients, and we identify the mutation. Then we characterize that mutation by putting or introducing and develop animal and see whether this phenotype is being seen there in those animals. That means that, yes, this is the mutation which is responsible for causing the disease. So that's the way we approach. Like if it is a mutation which has not been reported so far, how to come to the conclusion this is the mutation which is responsible for the disease. So we create animal model. We also try to treat that using different technology, and that would be useful for either the patients or in the future. Then convey all this information back to the patient or patient's family through our collaborators using telemedicine and so on. So that's a new program which we got in CDFD, funded by our organization, Department of Biotechnology. And that, I think that would go much more in a broader way. So this is a good beginning, right? So once we are successful in this, we show the successful of this particular program, there will be many more such programs evolving in the country to address some of the very, very important issues, which is at societal level. Okay, more particularly in the rural area, because that's where the endogamy and consanguinity is very, very high.
Thank you so much, sir. I think that's all the questions that we have. But any final comments that you would like to make, or any book suggestions that you have, please go ahead.
Yeah, my suggestion to public or those who are going to see this is that understanding the subjects or understanding whatever I've made in this program, that one would what can approach CDFD if they find some very unique disease which has not been known so far, they can always approach us, and we would able to do the genome sequencing, identify the mutation, and give appropriate counseling. And they can also spread this news to those others those who need this. Okay, so that way we can actually spread this information to many more people, and they would all benefit.
We would just like to thank you, sir, for doing this and accepting our invitation to be
No
the guest
problem.
at our podcast. Thank you so much, sir.
Okay, okay. I hope the information, whatever I conveyed, is useful.
Definitely,
Yes.
definitely. Because both of us are biology students, so it was a very
I-
insightful experience for us as well.
Okay. Okay. Wish you all the very best.
Thank
Thank
you
you.
so much, sir. Thank you.
Thanks for no making this. Bye.
Thanks
Send
for
me
accepting the invitation.
Okay. Okay. Bye.
Thank you so much.